Okay, I think we're recording. So welcome everybody. Thank you for joining us to our Zoom call today. We are delighted to share with you a very special guest who I'm going to introduce in a moment. I think you are absolutely in for a treat. We really appreciate you tuning in and all of your support of your friends and family members with Koolen-de syndrome that have brought you to this call today. I'm Casey, and I am once again going to be your host on behalf of the Koolen-de Syndrome Foundation. I am a cool mom, as I like to say, to a little boy named Hudson who just turned 11 and was diagnosed with KdVS about 5 years ago. And like many of you, I'm always very eager to see what research is underway to help provide more support for my loved one. I'm always honored to share the screen with our distinguished and very, very smart guests. And you are in for such a treat today because we are going to be introducing to you Dr. Brooke Latour. So Brooke, I want to say hello to you, give you a chance to say hello, and then I'm going to tell everybody all about how smart you are.
Well, thank you for having me. I am really excited to be here. This is actually a very special and meaningful thing to be a part of this. A lot of my background, I've done very fundamental research. So having the opportunity to work here with you and with the foundation has really been very inspiring and has really helped motivate a lot of our decisions and our research and, given us important goals in our research. So I've enjoyed it immensely, and I'm really excited to share our research with you today.
Well, we really appreciate you being here, and I do want to take just a minute to brag on you. I always just feel so thankful, again, as I said, as a mom to someone with Koolen-de syndrome, and when I see all of these really intelligent people that are dedicating so much of their work to helping us find answers, it's always extremely inspiring. I always leave these calls just really uplifted. So thank you. All will be impressed. Dr. Latour obtained a bachelor's degree in biology and economics at Oberlin College in Ohio. With an interest in scientific research, she went on to work as a staff research assistant at the Reagan Institute of Massachusetts Hospital, Massachusetts Institute of Technology, and Harvard in Boston, Massachusetts. She went on to the University of California in Los Angeles, where she discovered her interest in developmental biology. In 2012, she moved to the Netherlands, where she received her master's degree in biomedical science at the University of Utrecht. Am I saying that right?
Yes.
Close enough. Followed by a doctoral degree in neural development and disease in the human genetics department at the Radboud Medical Center. And for those of you who know Dr. Koolen-de, the Radboud Medical Center should ring a bell, which we'll talk about that more in a minute. In 2021, Dr. Latour was named the Koolen-de Syndrome Foundation Fellow for her work in neural development and neurodevelopmental disorders and how it relates to KdVS. Alongside her supervising researcher, Dr. Bert de Vries, Latour and a team of fellow researchers set off to explore how mutations that lead to KdVS give rise to altered brain development by creating cerebral organoids, or brain in a dish. This grant was made possible by the funds raised through the Foundation's annual Cool campaign. And oh my goodness, Dr. Latour, we were so proud. I think all of us who saw when we were able to raise the type of money to give you that honor, and we were so proud to call you our very first. It's just been really, really awesome. So you're like a celebrity to all of us who have been fundraising. I also want to mention today that this would not be possible, these types of Zoom calls and connecting with all of you, if it weren't for the KdVS Foundation and the commitment to educate, increase awareness, and promote research for the support and enrichment of individuals that are living with KdVS and their families. So if you want to take a moment after the call to learn more about the foundation and the work that we do, please visit KdVSFoundation.org, and you can learn all about our medical advisory board members as well as a lot of the research that's underway. Just a bit of housekeeping, and then we're going to jump right into the program. Our call is being recorded today, so we will be sharing the link after via email and social media, so please feel free to share that with all of your friends and anyone that may have missed the call. Also, I do want to note that you can see a Q&A button at the bottom of your screen. So as Dr. Latour is going through the presentation, if there are things that are coming to mind or something you're extra curious about or want clarification around, please just don't hesitate for a second. Every question is a good question. Just put it in the Q&A, and we will do our very best to answer any of those questions that are pertinent to her research and her area of expertise. So with that being said, let's not spend any more time on housekeeping. Let's just jump right in and get to know Dr. Brooke Latour a little bit better. So I want to just ask you a few questions we always like to ask our guests so the families and friends can get to know who you are as a person. So first, we'd just love to know, we heard a little bit in your bio, but what was it and kind of what was that moment where you said, I really want to do this research? You know, I'm really drawn to this field of science.
Yeah, that's a great question. So initially, I studied hematopoiesis, which is the development of the blood compartment of the body. So the part of the body that gives rise to the immune system. And I realized while I was studying that I was really interested in embryogenesis and the overall development of the human body. And the more I kind of researched it and gained background in it, I realized specifically the brain was what kind of held my interest. And this was kind of during the period of my master's. And then following that, I started a PhD in the human genetics department, also at the Radboud. And I studied a small organelle called the cilium. And the cilium can be thought of kind of as an antenna of a cell. And I studied it kind of in a really broad sense, like how it affected embryogenesis overall, the development of a variety of organs, including the kidneys. And over the course of that, I and others, of course, noticed that there were a lot of neurological phenotypes seen in individuals with disorders due to ciliary dysfunction. And so that kind of led me to focus in on brain phenotypes we saw in ciliopathies. And it was actually via that work that I came in contact with Dr. Nadif Kasri, my current supervisor. And we came up with the idea that I might set up a model in his lab to study developmental disorders. And in his lab, we of course study a variety of different epigenetic modifiers. And he said to me, I know you should do Cancel 1. You will really like Cancel 1. So that was actually how I got into studying Koolen-de syndrome. And so after I set up this cerebral organoid protocol, I guess he interacted with the foundation, and upon showing our preliminary results, we were able to get funding to do more extensive research and validate the results we had already obtained. So that was kind of how I got to where I am now.
That's so exciting. Yeah, we love talking to Dr. Kasri, and I know you all did some stuff for us last summer when we had our summit, or our virtual summit. But it's, you know, the work that you all are doing is so incredibly fascinating and interesting. And I'm not a science person, so I'm on the business side of healthcare. So I'm always blown away just by what you're able to uncover and what you've been able to learn so far. So I know everyone's in for a treat and I think especially even those that, you know, maybe a little bit newer to their journey. I think for those of us who've been on it for a while, it's always very interesting to hear what's coming and what's happened lately. But I think for the people who are very new to their Koolen-de journey, just to see how far you've come and what you all have already been able to learn, I think it's always very inspiring and encouraging for families looking for answers. So tell us a little bit about just you yourself. So let's take science out of the equation for a minute. Just tell us a couple of fun facts about yourself. You know, anything that maybe you like to do for fun or that might be interesting about you.
So I'm originally from Los Angeles, California. And growing up, both my parents actually work in the film industry. And when I told them I wanted to go into science, they were like, really? A lot of my friends that I knew in labs in the US, they came from science or medical background families. So my parents were excited, but didn't fully understand my passion. And so, yeah, that took me on a journey that they were really supportive of course, but that they didn't fully understand, or you know, when I said I wanted to move to the Netherlands to do my master's, they were kind of like, oh, okay. So I came here and I've been living here now for several years. I've lived in a variety of cities in the Netherlands. And I think, honestly, having had the experience of living abroad, I really think that enriches your experience of studying science, because, of course, in the US, there is a lot of funding for fundamental research. And that I think is great, but it can promote a different sort of environment for research. In the Netherlands, there is a little bit less funding, and therefore we're kind of forced to be more collaborative. And I think that works also very well with my personality. So it's been really fun to kind of network and create a situation in which we can just try to get as much done as possible for furthering our understanding, but also furthering what we can do for the patient. Living in well, I guess actually studying at the Radboud has been great because there's this really close connection between research and the clinic. I realize I'm now venturing back into the science realm and not the personal realm.
That's okay. Yeah. they're two mixed together, right? It's hard not to.
Honestly, yes, they kind of are. Yeah. Yeah.
And you've stayed in I mean, I think what I read earlier is that you've been in the Netherlands since 2012. Is that correct?
That is correct.
Which is a long time. So, I mean, you must obviously enjoy the work and living there to have stayed that long, which is really, really interesting. So I guess last question before you jump into your research, let's stay on the Netherlands for a second. So those who, again, are new to KdVS and those of us who've been around it for a while, we hear the Netherlands and we all instantly think, oh my gosh, all of our rock star KdVS researchers live there. So what's something, kind of an interesting fun fact you can tell us about the Netherlands so that when we're imagining all of you rock stars living over there, we can kind of think about it when we're all back here in the US?
Well, actually, so the Netherlands, a lot of the cities have a lot of really beautiful canals. And the Dutch were actually the first ones to build and design New York City. So that is why New York City is on a grid system. And if you go to Amsterdam, you'll see that the layout is very similar in terms of how you can find different areas. You have concentric streets and canals. In Nijmegen, unfortunately, we don't really have any canals, but we have two large rivers, which Nijmegen falls in between. It is an incredibly beautiful country to visit, and it's very highly populated per square kilometer. So wherever you go, you will never be in the middle of nowhere. Yeah, it's if you come here, I would highly recommend not only seeing Amsterdam, which is of course the hub you would probably fly into, but also seeing Utrecht. It's also known as the church city. I'm honestly not exactly sure why, because there are not that many churches, but there is some really beautiful old buildings that are worth exploring. And yeah, it's a really amazing place to visit.
Sounds beautiful. I think, and we need a KdVS field trip to the Netherlands,
yes.
I think.
Oh, that would be great. And I know for a fact that everyone here would be very happy to host you.
That would be pretty cool. Well, without further ado, I really appreciate you spending a little bit of time letting us get to know you better, but I'm very excited to get into your research. So I'm going to let you just go ahead and share your screen and take it from here and let us know some of the amazing things you've been up to. And then again, just a reminder to those watching, please feel free to put your questions in and I'll be sure and monitor those as we go along. Perfect. I can see it.
Okay, great. And you can see the proper screen, not the presentation screen?
Yes. I'll double check when you click forward, but right now it looks perfect.
Okay. So now I would like to share our data with you on work I've been doing over the last year and a half, modeling Koolen-de syndrome and cerebral organoids. So to start with, I'm going to give a brief introduction on Koolen-de. We know that Koolen-de syndrome was initially characterized as a microdeletion syndrome. It was identified in individuals who had a microdeletion on chromosome 17 in the area 2131. And that area encompasses these 5 genes that you see here, and was later resolved to being caused by variations in the gene cancel1. And what they found, clinically speaking, is that variations in cancel-1 resulted in a very similar phenotype to what we see in individuals with the microdeletion. And this neurodevelopmental disorder is characterized by intellectual disability, variable manifestations of language and speech delay. Early on in childhood, you see some instances of hypertonia. There's the distinctive friendly social behavior. There are also variable manifestations of epilepsy and and a heterogeneous presentation of congenital defects. So when we think about the brain in Koolen-de syndrome, there actually has not been an extensive clinical analysis of this. There have been some reports, of course, also done in Nijmegen by Dr. Bert de Vries and by Dr. Koolen, but it hasn't been with a huge number of individuals. So we do know that what they observe to variable degrees is malformation of the corpus callosum. The corpus callosum is the axonal tract that connects the two hemispheres of the brain. We know that there's abnormal formation of the hippocampus. This area is responsible for learning and memory. And we know that there are dilated ventricles. So that means basically you have enlargement of your brain ventricles. And there can be a manifestation of heterotopia. So this is a neuronal migration disorder. And then there are some like brainstem and cerebellum abnormalities are sometimes observed. So we know that there's a lot of variation, and we don't know exactly what the consistent manifestation is, what is kind of more individualized, and what is happening as a result of cancel-1 disruption. And so we are using the cerebral organoid model to try and figure that out. And therefore, I think in order to kind of get into that data, I would like to first take you through brain development. And so we can later reflect back on potentially what cancel-1 is doing during that time. So this will be a little bit of an abridged explanation, but I'm going to try to take you through it in a structured way. So human brain development occurs in specific but overlapping stages, and these stages include proliferation of your neural stem cell population, followed by differentiation of this population, whereby you get neurons and other neural cell types. Once you have the differentiation and formation of neurons, these neurons migrate to where they need to be within the brain. And then once they've migrated to their appropriate location, they actually connect to each other and form neural circuits. So first, to discuss what neural stem cell proliferation means. So basically, what you have is you have a few neural stem cells, and they replicate themselves. And this type of replication, this cell division, is autoreplicative. So the cell literally divides into two cells that are replicas of the original cell. And it does this to create a large enough pool of cells that are neural stem cells that can give rise to the entirety of the brain. This is actually very much like on the onset of pregnancy, you have an initial fertilized egg, and then this egg becomes 2 cells. These 2 cells divide and form 4 cells, and then 8 cells, and so on. But these original stem cells that give rise to the developing embryo, they need to expand enough to create enough cells to create the entire human body. So on a smaller level, we're seeing this exact same process with neurodevelopment. So after we have firmly established this important stem cell pool, and its size, it's large enough to form all of the cells in the human brain, we see a process called differentiation. And these cells, when they divide, they no longer form 2 replicas of the same stem cell, but they form more differentiated cells. So cells that are more committed to the neural fate of being either neurons or something called intermediate progenitors. Intermediate progenitors are not quite yet neurons or other cell types in the brain, such as astrocytes, but they are further committed than these initial neural stem cells. And what we see with this differentiation is as time goes on, there is this development in this inside out fashion. So you're number one, you see the VZ, this is the ventricular zone. This is basically the niche for neural progenitor cells. Then after the formation of this niche, where you have the expansion of your neural progenitor population, I use the word progenitor interchangeably with stem cell because it is the same thing. I just want to be clear on that. So after you establish this stem cell niche of neurostem cells, you then develop the subventricular zone, so the number 2 layer. And this layer is populated by intermediate progenitors. So again, these cells are committed to neuronal fates, but they have not yet fully formed into neurons. And then after that, you have the formation of neurons. When these neurons are first born, they are born within the subventricular zone, and then they migrate out to the cortical plate. You see the number 3 here, CP, that's the cortical plate. And then they take up residency in this area so that they might establish connections with the other neurons in the cortical plate. So I'm going to sort of use this as an example where I say individual neurons are a lot like individual computers. They have a lot of computational power, but once they form connections to each other, they develop a greater computational capacity. And these connections are formed literally through physical interaction. And we can measure that interaction via measuring the electrical activity that these cells communicate with each other with. So literally these connections form the capacity for these cells to talk to each other. And what you're seeing here in this graph, each line represents, so each horizontal line represents an individual neuron that we're monitoring, and each tick, so vertical line you see here, represents an electrical blep, or action potential if you want the official terminology. And we can see that these neurons are firing spontaneously, but if you look right here at this line where they all seem to be firing at the same time, this shows that they're actually synaptically connected and communicating with each other because they're all responding in the same fashion. And what we see over time is that these networks become more complex and there's additional communication. And also this communication is more developed. So initially you see lots of these individual random spikes and only one or so what we call network bursts, so proof that these neurons are connected. But as time goes on, you see the communication becomes more organized and you see multiple network bursts, so multiple instances of intercommunication, and it's kind of at regular intervals. So that's how we can establish maturation of neural circuitry. So this is just a very brief overview of the stages of development of the human brain. But the basic take-home points from these few slides is that initially we have a neural stem cell population that undergoes self-replicative division to establish your neural stem cell pool. And this is a stage we call proliferation. And then once this stem cell pool is sufficiently expanded such that it can populate all the layers of the cerebral cortex, we start to have differentiation. So these stem cells start to become more committed to neuronal fates, to forming neurons and other cells that you find in the brain. And once these neurons are born, they can migrate to the appropriate location within the brain and set up residency, so establish where they need to be. And then they extend their processes to connect with other neurons. And in this way, they're able to communicate with each other. And this is what we call neurocircuitry formation. So all that being said, how do we look at that in KdVS? Well, we are extremely lucky because we have a relationship with the KdVS Foundation. And because of that, we have access to a lot of patient lines. But before I actually go into that, I would like to kind of talk about what a stem cell is, because I realize that's not common language. So iPS cells stand for induced pluripotent stem cells. And what that means is essentially a terminally differentiated cell, so what that is maybe a skin cell or a blood cell, can be taken and reprogrammed into an embryonic state. And that embryonic state allows it to be differentiated into any cell type. So in our case, we differentiate them into neurons or cerebral organoids, but really they can be used to study any sort of tissue or affected tissue in specific diseases. So this actually amazing technology was developed by someone named Dr. Shinya Yamanaka, and he won the Nobel Prize for this in 2012. And he discovered a set of factors that could be used to reprogram skin cells or blood cells or other tissue types into embryonic stem cells. So using these embryonic stem cells from patients, we can make and study neuronal development. And this is really important because, as I mentioned earlier, we see quite some variability in the manifestation of neural phenotypes in KdVS. And therefore, we can start to look at both differences between individuals, but also convergences of phenotypes. And then we can kind of get a sense of where we can target for potential treatment intervention. So we in our group that study KdVS, we use essentially these, the cell lines you see here on your screen. The empty box indicates an unaffected male, the empty circle indicates an unaffected female, and then the filled circle represents an affected female. So we have 3 patient lines. We have a maternal control. So one of the patient's mothers gave us cells to act as a genetically related control. From this control line, we were able to create a genetically edited line using CRISPR-Cas9 technology. And then we also have 2 independent control lines. For today, I will show you data from these 3 specific lines. So the affected patient, the control line derived from the mother, and then the CRISPR edited line. So in order to appreciate, I think, fully what organoids are, it's important to understand what they are. So organoids can be of any tissue. They can be kidney, they can be heart. Today, everything I will be talking about will be specifically cerebral or brain organoids. But they all have a common definition, which is they must be stem cell derived. They must be self-organizing. So this picture in the background you see here, this is an example of what I mean by self-organizing. You can actually see these flower-like structures. The cells, you add a common kind of recipe of factors to these cells in their medium, and they know to form these flower-like structures. These structures are kind of recapitulating the formation of the neural tube. And they also, as they start to grow and differentiate, they form their stem cell niche here, and then the more differentiated cells actually form around the edges. So that's what's meant by self-organizing. And then these cells must undergo spatial temporal lineage restriction in a manner similar to the organ it represents. So what that means is basically what I was saying earlier when I talked about proliferation, differentiation, migration, and then circuitry formation. We see that over time, these cells become more committed to the fate that they will end up. So they go from being stem cells all the way to being neurons. And this needs to happen in a fashion that is comparable time-wise to the development of the organ during pregnancy. And then, of course, in that same vein, the organoid needs to contain the cell types of the organ it resembles. So in our case, with the cerebral organoids, we need to see stem cells that are neural. We need to see intermediate progenitors. We need to see neurons, and we need to later see astrocytes, which are support cells for these neurons. So how do we make organoids from iPS cells? This may seem like a very simple question, but it's pretty fundamental to what we do. So you can think of actually a lot of the things we do in the lab like baking. We have a recipe book, which we call a protocol, which often involves some tweaking and modifications, much like a recipe would, depending on your personal taste. And yeah, we go through these stages or these steps, and we add these different ingredients, and we get, hopefully, the model that we're looking for. So to begin, we start with 9,000 iPS cells, so 9,000 stem cells, and we plate them in a U-bottom shaped well. So this is a dish that is literally shaped like a U, but will not allow the cells to stick to the bottom. So in that way, they collect and form this ball-like structure you see here, this sort of spheroid of cells. You can see it specifically here. These are the cells, and then in the schematic here. And then after, on day 0, this is what we do. And then after 5 days of supporting the growth and formation of these spheroid structures, we start to add factors that force them to the neural lineage. So during this spheroid formation, all we do is support cell growth. We don't add any factors that force them to become neurons. They're just supported, and they're just proliferating. Then on day 5, we add inductive factors. So what that means is they're now being forced towards neural ectoderm, which is really the earliest stages of brain cells. And we see that they actually start to take on this polarized structure, and they form these kind of flower-like structures like you saw in the picture previously. And it's a little bit harder to see here, but that's also what's happening. There's kind of these folds you see in this cerebral organoid. We then provide certain proteins, which are called extracellular matrix proteins. And these actually really serve as support proteins to allow these cells to kind of expand and form these polarized structures. And we start to see that they take on a more developed flower-like shape, and they also start to have these what we call luminal spaces within the rosettes. And these luminal spaces are reminiscent of the formation of a human brain ventricle. So what you're seeing here, for example, with these 3 rosettes, this is actually what you would get if you had 3 neural tubes forming together. So it's not a perfect picture of neural brain formation, but it does give us insight to what's going on here. And then we add maturation factors, and we start to see that within our stem cell niche here, they start to form intermediate progenitors, these cells that are more committed to the neuronal fate. And as more time goes on, we see that they start to form terminally differentiated neurons. And again, we have this inside-out formation, much like you would expect with brain formation, where we have the ventricular zone, which is the neurostem cell niche, the subventricular zone containing the intermediate progenitors, and the cortical plate, which contains the terminally differentiated neurons. In other words, this is what it looks like in practice. So these are actual cerebral organoids shown at various time points. At day 5, you see this kind of spherical-like structure. The cells have not taken on any sort of polarized shape yet. By day 10, well, actually by day 7 through 10, we start to see the formation of these, what are called neural epithelial rosettes. These are these flower-like structures that contain neural stem cells. And then we just see a continued expansion of this up until day 60, where we see here is the stem cell niche. Here is the intermediate zone, or the subventricular zone with the intermediate progenitors. And here is the cortical plate containing neurons. So what I would like to actually also explain with this slide is that you can see that there are various colors associated with the various regions. And we have a technique, it's called immunofluorescence. And what that technique does is we know that these specific cells express certain proteins that define their identity. So for example, stem cells express different proteins than intermediate progenitors. And we can stain these proteins with different colors so we can see them and we can identify which cells are which cell types. So, for example, in red here, you can see that these are progenitor cells. And in green here, you can see that these are neurons because we've stained them for our neuronal specific marker. So the fact that we can do this is really exciting, but it becomes really important to know what can we do with this? Like, it's very great that we can kind of mimic brain development, but what sort of assays can we do to gain insight into what's going on in brain development in individuals with KdVS? So something very important to mention about stem cell proliferation and differentiation is that we can actually tell whether a cell is going to decide to be proliferating or differentiating depending on the orientation of that division. So here you can see cells undergoing division, and they're doing it in 3 different ways. So with vertical division, this is the self-replicating proliferative division. Oblique division and horizontal division both occur with differentiation. So this is when the cells start to commit to a certain fate after they've gone through their proliferation stage. And if you look back here at this organoid slice, you can see in white here, we've stained the neural progenitor cells that are undergoing division. So we can actually zoom in, shown here in this diagram, and look at the type of division that these cells are undergoing. And in that way, get a sense of what stage they're in, whether they're still in a proliferation stage or they've already switched to a differentiation stage. And what we see when we do this is something that is somewhat expected with the control. We see the majority of divisions at an early time point. So for us, this is day 10 of organoid development. We see that this replicated division is expanding the neural stem cell population, much like you would expect. And there is a limited degree of oblique and horizontal division. So you have early stages of differentiation, but the majority of divisions are really these self-replicative proliferative divisions. However, when we look at the same time point and we look at the KdVS affected patient, as well as the CRISPR'd or genetically edited line, we see that there has been a somewhat premature switch to this differentiation division. So there's less proliferation and there's more differentiation at this early stage than you would expect. Now, what could this mean? So we're not entirely sure yet because we've investigated a few avenues and it's not entirely clear. However, I will say that it could definitely mean that there is a smaller supply of stem cells to create other neural cell types in the KdVS affected organoids. So to kind of address what this could mean with further differentiation, we first looked at a stem cell population that resides in the ventricular zone, so the stem cell niche, and the subventricular zone, which you can think of as the intermediate progenitor niche. And what I was talking about earlier with immunofluorescence, we can stain a specific protein called HOPX, which marks and identifies these specific cell types. And this is really useful because we can see the cortical plate, the subventricular zone, and the ventricular zone here in these 3 different layers. And we can identify how many cells are located within each of these zones within the control and compare it with how many of these cell types we see in those zones in the KdVS affected patient. And what we see when we look at the CRISPR line and the patient is there is a significant reduction in this progenitor population within both the ventricular zone and the subventricular zone. So we know that these cells give rise to intermediate progenitor cells. These cells, again, you can see here in this schematic, they're in yellow, located in the subventricular zone for the most part. And these are cross sections of this developed neural epithelial rosette. So you can see the cortical plate, again, the subventricular zone and the ventricular zone. And again, we see a significant reduction in TBR2 positive intermediate progenitors. Now, something I didn't mention is there are different types of intermediate progenitors, and this TBR2 marks a very specific type. And we know that this specific type gives rise to excitatory neurons. And the reason we're specifically interested in excitatory neurons is because we can directly measure the electrical signal, so the circuitry formation from that specific cell type. So here you can see, we see a reduction in the KdVS affected organoids. And what we also see is that there's a reduction in the specific excitatory neurons that they give rise to. Now, this may seem very straightforward and like, well, obviously that's what you would expect, but it was very significant that we could actually observe this in process over the course of development, because as you can appreciate, this is several months of changes in cell types and development. So what we see here is in this diagram, again, this is the cortical plate. You see SETB2, which is an upper layer neuron marker. So these neurons develop a little bit later than the CTIP2 positive neurons. These are excitatory neurons that are a little bit deeper. But we do know that TBR2 cells give rise to these neuronal subtypes, and we do know that both of these populations are affected by the reduction in stem cells and intermediate progenitors. So what we would expect to see as a result of this reduction is differences in KdVS neurons' abilities to form neural circuitry. And when we measure this, when we measure the electrical communication between these cells, we do indeed see this. So in the top panel here, you can see the control, and we can see that there are defined network bursts. So these are instances where you can see that all the neurons are firing together because they're synaptically connected. Whereas here, you can see they are synaptically connected, but it's to a much lesser degree. And therefore, what we see is that these networks seem to be less mature, less developed than what we see at a given time point for the control. And so this is quantified here. You can see the control gives you a certain number of network bursts. So these lines here per minute. But the KdVS patient and CRISPR line are significantly reduced when compared to that. And then what we also see is an increased percentage of random spiking. So random spikes are these individual ticks that you see, and they occur when a cell is firing an action potential, but is not coordinated with the other cells. So these are more stochastic events of individual neurons firing action potentials. And this is a sign of a younger neural network, so a network that is not completely synaptically connected yet. So all this information is of course very interesting, and we're still doing some ongoing research to investigate it more fully and see why we descriptively see some of the things that we see. But what we can know is that there are certain aspects that we can, or certain areas we can intervene to kind of ameliorate aspects of disruption. So this is work done by my colleague, Dr. Koolen-de, in a paper that was published, I think early this year actually, where she showed that in 2D, you can add apocynin, which is an antioxidant, to these neurons, so to these excitatory neurons, and you can kind of restore or decrease the effect that you see. So you can see here that, again, there are less network bursts, so these synaptically connected instances, but with the addition of apocynin with the CRISPR line, so the genetically edited line, you see that network bursts are indeed occurring, much like what you see in the control. And this is a graphical representation of these plots. So you see without aposinin, there's a decrease, but with it, it's restored to almost control levels. And then again, you see here, there is a decrease in random spiking with the addition, but it's not as significant as the restoration of network bursts per minute. Now, I must say that this is a two-dimensional culture only containing one cell type. So while this is very promising, it's also a very reductive model. So we're trying right now to recapitulate this with the organoids, as the organoids have multiple cell types and are more similar to human brains in that they contain all the cell types of the human brain, and they sort of develop in a more similar fashion. So it's a less artificial system. And we have indeed seen that the synaptic connections are similar to what we see in the 2D model, but we have yet to perform the treatment experiments to see if indeed it works the same way. So in summary, basically what we find is that the earliest population of neurostem cells are affected in KdVS organoids, and this leads to a decrease in the differentiated intermediate neural progenitors and the subsequent formation of these excitatory neurons. And this data suggests that early progenitor proliferation and differentiation are affected in cases of cancel-1 haploid insufficiency. And this change in developmental trajectory could underlie the changes in circuitry maturation that we observe in the 2D cultures of neurons. But we're currently working to see if we can recapitulate this data with the organoids. So I hope that in presenting this data to you and kind of the paradigm within which we work in the lab to kind of use KdVS specific stem cells to make brain organoids, and then kind of learn more about the mechanisms of brain development with this organoid model may inform potential treatment strategies, which we can then return to the patients. All of this is, of course, in its very early stages, and it's just research that we're doing, but it's definitely what we are working towards. So with that, I would like to thank all of the people involved in this research, the KdVS Foundation, Neil, Bert, and all the people in the lab who have helped me with these assays and to set up the cerebral organoid protocol. And thank you for your attention, and I'm really happy to take any questions you might have now.
Well, that was amazing. Like I said, I always learn so much, and it is hopeful. I mean, what you're able to learn and able to see. We do have a couple of questions, and again, I just want to remind our viewers we do have about 12 minutes. So if there are things that are on your mind, please don't hesitate to put them in the Q&A, and we'll be happy to take your questions. So the first one comes from Blake, and Blake is asking, does oxidative stress, like increase in mitochondria or byproducts or lower intracellular autophagy, as shown in KdVS cell lines or cells, induce differentiation in early neural cell development?
Oh, this is a very interesting point. So we in our group have investigated its role, of course, as you seem very well informed on, in neurons. And most recently, a few months ago, a paper came out looking at this in the context of the cancel-1 mouse. And they showed that cancel-1 affected expression of specific autophagy-related genes. In the mouse paper, it was, I believe, STIX17. And this affected autophagism formation. So I think that increases in ROS would hypothetically occur also in your progenitor populations. It would not just be restricted to neurons, and therefore could indeed have an effect on the state of the cell. We have not specifically looked at this, but that is actually a very interesting and important point. And hopefully I can follow up eventually with that answer more specifically.
Yeah, that's a great question. I know all of our families and friends, I feel like are so well researched and always ask such good questions. We have another really good question here from Amanda, wanting to know how near in the future are we from testing on the organoids?
So actually, I assume you mean testing, for example, these antioxidants and other treatments. Actually,
Yeah.
I am working right now with Dr. Koolen-de, who is the first author of the autophagy paper, and with Neil, and we are setting up a protocol where we can do a slightly higher throughput screen where we look at the organoids on these multi-electrode arrays. So we have these high density multi-electrode arrays, which contain electrodes, which we can plate the organoids on and measure their synaptic firing. And based on that readout, we can start to add compounds to them relatively soon. We have, for the most part, optimize the plating the organoids on MIAs. It's a little bit more complicated than the 2D system, but now that that is up and running, we could start that within as soon as 3 to 6 months, actually. So
Wow.
that is the next stages of what we're planning to do, indeed.
That's really exciting. Are there any other questions we have that are specific to Dr. Latour's research, or anything else you all are curious to know about? These are good questions. Dr. Latour, are there any questions that you've heard families ask you before that you are surprised haven't been asked? So maybe we've got people out there that aren't even sure what to ask.
So I must say, first of all, that fortunately, of course, I got into this research, but unfortunately it occurred during the time of Corona. So my first year of KdVS research was really during 2020. And as a result of that, I haven't had as much interaction with families as I would like. And I'm actually very excited to do that in the coming years. But a question that Neil actually told me that he often gets is, if we see a disruption so early on in brain development, even before we know we have a diagnosis, what do we do? How do we deal with that? And the answer to that is, I don't know, but I do know that there are many aspects that we can learn from this early change, and therefore will give us insight into aspects that we can target with treatment. For example, neurocircuitry, formation and maturation, and that can be very informative. So if we know what's going wrong or what's happening early on, this will allow us insight to predict targets for treatment and potentially ameliorate some of the symptoms that we see.
Yeah, because I think that's, as I think about that as a parent, those are the things that are always fresh on my mind, and now my son's a little further in his journey as an 11-year-old, but I look at just the evolution just in 11 years. I mean, we couldn't even get the genetic testing. I mean, quite frankly, it wasn't even affordable when my son was born. But, now just the evolution even of genetic testing, where families are learning so much sooner that they have KdVS, and you think, gosh, with what you all are able to learn, are there ways, like you said, with the drugs, or are there ways that we could correct? I mean, I think that's what is on all of our minds. When patients or these families are diagnosed early on, will there one day in the future, be a way to correct some of those, abnormalities or misfirings or lack of connectivity, I guess. It's something that I think we're all really hopeful about.
Yeah, and I honestly think I can say that we as researchers are also very hopeful. We found specific elements that can be, intervened with, and therefore, lessened or treated to a certain extent, making things within our systems more manageable, or restore them to something that is similar to the control. And therefore, we are hopeful that we can kind of continue to learn more about mechanisms and targets, and therefore use specific compounds to restore things, basically.
Yeah, it's really incredible. We have another really good question in here from Mohammed asking, have you seen any change in proliferation, cell cycle exit, or cell fate choices in neural stem cells as a result of these mutations? other than a reduced number of IPs?
So so all the data that I showed you today was data that we have extensively validated over the last year. We've run these experiments multiple times, and we see the same thing. I do have some newer data where we look at cell cycle exit, and we do see that cells, so we use both the iPS cells and we look at the neurostem cells, and we see that it seems that with cancel 1 haploid insufficiency, there's a stall in the G2M phase of the cell cycle. And what that means is basically there could be an ongoing DNA damage response in these cells. And that is causing a delay in cell cycle. So it delays entry into mitosis, which is the phase in which you see these cells coming apart. So yes, we do see that, but this is very early data. We do see it consistently with the iPS cells and with the neuroprogenitor cells in the organoids. But we're beginning some work with a group in Leiden who specializes in DNA damage response, and we hope to learn more about what's going on with their help.
Wonderful. Let me look and see if we have any other questions before we have to wrap up. We have one more. Let's take one more, if that's okay with you. Does this research correlate with a high prevalence of epilepsy with KdVS, and in particular, the abnormal EEG findings, even when no clinical seizure presentation at the time?
So I have not looked into that, actually. We are trying to establish a way of assessing epilepsy with organoids. We don't have very much experience with that with the 3D model. There are people within our group who look at that in the 2D model with other disorders like Dravet syndrome, for example. So unfortunately, I cannot answer that question now, but it is a really interesting and important question, and hopefully, a direction we are attempting to take, and hopefully something that we can have more information on in the future.
That's wonderful. We're going to have you back a whole bunch to tell us all these.
Thank you. I really appreciate
all these incredible things you're doing, because I mean, it feels like the path you're on and what you're able to learn and the answers that we're going to be able to find. It's very promising. I know we all try to, not get too excited, but yet it's hard not to when you hear these types of things. So we just really appreciate you sharing this great information. I think I would be remiss if I didn't mention, especially with you being, our first fellow, that, right now we recently had KdVS Awareness Day, and we are right now in what we call our Cool campaign, which is where all the families work hard together to fundraise every year. And that's what gave us the ability to fund your particular research, which is giving us all these great answers. So I do want to encourage those of you that are on today just to share this. We'll get the recording out as soon as possible, and share this with your friends and your families and your communities, because the more we continue to fundraise around this, the more we're going to be able to do to support Dr. Latour's work. So it's very good timing that you're sharing all of this great work that you all are doing. So again, just really want to thank you for taking time out of your busy schedule to share your research with us and to educate all of our listeners. Want to thank those of you that are on the call for taking time out of your busy day or evening, depending on where you're located around the world, to just spend time with us. And want to thank the foundation for continuing to host and sponsor these events so that we can continue to educate our friends and families. So again, many thanks to all of you. We hope that you'll continue to tune in as we do more of these webinars with our very distinguished and very important guests. And just hope that you all have a wonderful day. Dr. Latour, thank you again. So appreciate you being here.
Oh, you're welcome. And thank you so very much for having me. And thank you for all the very well-informed questions. The autophagy question has inspired an experiment idea. So thank you for that.
Wonderful. Yes, such good questions. So thank you again, everybody, and we'll look forward to talking to you soon.
Okay. Thank you.
Bye-bye.
Bye.